So this was the process of ovulation, which we discussed before, that how this ovum is expelled out and it is taken up by the fallopian tube, and then and initially it is surrounded by a bunch of cells, which are the granulosa cells known as the corona radiata. And when it is fertilized by the sperm, the zygote is formed by the completion of the second meiotic division. This is the formation of a zygote, fusion of a male and a female pronuclei. When the sperm enters, the head and tail enters inside, and the head swells, which contains the DNA, and the two nuclei, one is the female pronucleus and the male pronucleus, join to form a single nucleus containing 46 chromosomes, and this cell is now a zygote. Right? And the zygote is now surrounded by just a thick membrane, this green layer. That thick membrane is the zona pellucida. Right? These corona cells, they will now leave the oocyte, and only the zona pellucida is surrounding, which is a very thick membrane or a sheet which is protecting the zygote. Now, soon the process of cleavage start, which takes 30 hours. So this step is after 30 hours. So this traveling or distance is 30-hour difference. And the whole of this process, it passes and reaches the uterine cavity. It takes 3 days. Right now, this within 30 hours, the zygote will start dividing, and the division is by the process of mitosis. And now, first mitosis, this will lead to 2-cell formation, then there will be 4-cell formation, 8-cell formation, and then 16-cell formation. And see what is happening. The size of this mass is not increasing, only the number of cells is increasing. And this zygote is these cells are surrounded by zona pellucida. This tough membrane, zona pellucida, keeps the size same. To increase the size, this zona pellucida must disappear. Right. So the zona pellucida, why the zona pellucida is present in the uterine tube? Because we don't want to increase the size, because the uterine tube is very thin. If the size keeps on increasing in the uterine tube, it may cause the burst in the uterine tube. So this zona pellucida has to be there so that the mitosis keeps on, but the size does not increase. So there will be smaller number of cells, and there's a compaction process going on. Now this is known as 16-cell stage, is known as morula. This embryo is morula, right? And this, when it enters the cavity, now this zona pellucida is digested by the secretions of this uterine cavity, and the fluid in this uterine cavity will seep in. Now this will seep in, and the fluid will accumulate in between the cells, and the cells, they will arrange themselves into groups. The cell number is increasing, and they are arranging themselves into groups. ये वो cells जो थे एक पूरा bunch बनाया हुआ था. Now they are arranging. As the fluid moves in, a small cavity is created inside, and that cavity fluid is known as the blastocyst cavity. And the cells are arranged into groups. One is forming the outer border, and the other one is in the form of a compact mass. This is blastocyst. Now you can see the size has increased, number of cells is increasing, and the size is very small. The number of cells is tremendous now. One layer of those cells have formed the outer boundary, and the other one are being collected at one end of this, right? So this is the blastocyst with the blastocyst cavity. This is now 4th day of the embryo. On 4th day, this is the it is in the uterine cavity, and the embryo is known as blastula. This was morula, and this is blastula. And now this embryo or the blastocyst will come and attach to the inner layer of the uterine cavity, which is known as the endometrium. You know this about it? The inner layer of the uterus is known as endometrium. This middle is the muscular layer, the myometrium. The outer thin connective tissue layer and the peritoneum is the perimetrium. This blastocyst is going to erode this and embed itself in the endometrium. This is the implantation. It digs in and it is embedded in. It's just like you just embed or you dig in the seed in the mud. आपने कोई प्लांट लगाना है or you want to have a small tree to be grown in it and you want to use the seeds are embedded in the mud, right? And the same thing will happen now here. When it comes in contact with this layer, now this will be eroded and it will be embedded within this wall, and this is the implantation process. Now this duration is 3 days. This is the 4th day. It floats for another 2 days, then gets attached on the last day of the week, 6th and 7th day, to the wall, and by 8th day start embedding. That is now in the 2nd week, it gets embedded into the wall. Right? So the first week of development is actually this traveling through the uterine tube, and then it will get a change into the blastocyst. These are the magnified pictures of the division of the zygote, the 2-cell stage. This is the zona pellucida. And the zygote has started dividing, and the first mitotic division completed formation of 2 cells. Each cell is known as blastomere. Can you read here? The blastomere. These are all blastomeres. These are all blastomeres. And the mitosis goes on. And can you see how this covering of the zona pellucida is forming a shell, just like the shell of the egg? And within this, there are smaller and number of cells. They're very small cells, but the number has increased. And this is the 16 cell stage, which is known as marula. And now when it has entered the uterine cavity, you can see the color of the zona pellucida is very light. It is being digested. It is degenerating. And ultimately, there is no covering here. Right? So this, when it thins out, the uterus, the fluid moves in, and this is now blastocyst cavity. This fluid will accumulate in between, and the cells will align into groups. The one type of cells will form the outer border, the other cells will collect on one end of it. This is known as this collection of the cells at one end is known as inner cell mass or the embryoblast. And this, which is forming the outer boundary, is known as outer cell mass or trophoblast. This is the next stage in which the blastocyst has increased in size, and this is the blastocyst cavity. The outer layer of cells is forming the outer boundary, outer cell mass, also known as trophoblast. I'm going to write it on the next slide, and you can then read it, what is this known as. And this is the inner cell mass, which is known as embryoblast. Actually, this outer layer will form the membranes and the placenta, and this inner cell mass or the embryoblast will form the embryo, the future embryo, and this is the blastocyst cavity, which is going to form the primary yolk sac. So the process of cleavage, which is actually the rapid mitotic divisions of the zygote, and as the mitosis occur, the number of cells increase, and these cells are known as the blastomeres. And this cleavage process starts 30 hours after fertilization, and cleavage process occurs within the uterine tube. And within this, the compaction process occurs, and there's no increase in the size because of presence of the zona pellucida, and this is important. Otherwise, this will lead to the burst of the uterine tube. Then comes the marula. We have discussed this, the marula formation, which is a 16 cell stage, and I should write compact mass of cells, and it is actually 16 cell stage. This is so popular, the 16 cell stage word, because this can be asked in the MCQs. Then you will be lost at what is the 16 cell stage. It's the marula, right, which is formed on the third day of fertilization, and it is within the uterine tube, and then it enters the uterus, and when it enters the uterus, it changes into the blastocyst, and entrance is on the fourth day in the uterine cavity, the uterus, right? And the spaces when they appear within this marula, that will lead to formation of a cavity, and the blastocyst is being formed. Now this blastocyst, the cells are the same blastomeres, and they are grouping into two groups: the outer cell mass known as trophoblast, and the inner cell mass is known as the embryoblast. ठीक है जी. So these were the two new names which were added in this, that this is the almost the end of the first week, but still one step is left, which occurs on the last day of the first week, that is on the seventh day. The initially the blastocyst was formed containing the inner cell mass, embryoblast, and outer cell mass, the trophoblast, which you have seen here. This is the embryoblast, and this is the trophoblast. And now what happens is that this blastocyst floats for 2 days after forming embryoblast and trophoblast, and when the zona pellucida completely degenerates, its size is increasing, and it will ultimately get attached to the wall of the uterus. This is the same blastocyst, which yellow is the outer cell mass, which is trophoblast, and this blue is the inner cell mass, which is embryoblast, and on one side, which is the embryonic pole or embryonic end, and the other end is known as abembryonic end. The embryonic end, this is known as embryonic pole. Why? Because the embryoblast is on this side, embryo will develop on this side. So it will attach to the wall of the uterus. This is the wall of the uterus, which has been magnified. I'll show you this diagram again. This is the uterus here. This is the embryonic pole, and the wall of the uterus is this one. It will attach by its embryonic pole to the uterine wall. This is the same attachment here. And this dipping in is the dipping in of the uterine glands. And now in this case, you can see this dipping in the uterine gland. This hole is the endometrium, and the blastocyst with inner cell mass. This is inner cell mass, and the outer cell mass, it is attached to the endometrium. The outer cell mass has the capability to attach, which is the trophoblast. So the trophoblast will attach itself to the uterine endometrium, and soon when it attaches, the cells of the trophoblast in this region proliferate, and they form finger-like extensions, and they form another layer of cells which are the on the outer side of this main trophoblast. Did you get this point? This is the trophoblast, and now the trophoblast at this end, at the embryonic end, shows proliferation, and it forms number of cells which grow in the form of finger-like extension. These are the finger-like extension. Can you make out the finger-like extensions? So there are two groups of the cells of trophoblast, one forming the finger-like extension, and the other one which is continuous with the rest of the trophoblast. These are now two groups of the trophoblast at the embryonic pole, and these two groups are now named as the syncytial trophoblast and the cytotrophoblast. The trophoblast now is in two groups at this end. These finger-like cells are known as syncytiotrophoblasts. This is very important. And the rest of the trophoblast, which is in yellow all around, is known as the cytotrophoblast. So the trophoblast is now in two parts by the seventh day, and it is when it touches and gets attached to the endometrium. The finger-like are syncytiotrophoblasts, and the rest is the cytotrophoblast. These finger-like cells actually dig in, and they cause digging and drag the blastocyst inside the endometrium. That is what is required. Another layer where the inner cells of the embryoblast, they flatten out and they form a thin layer of cell which is known as hypoblast. This is by the end of the first week. So the first week ends by this. The first week traveling through this is the day one when the zygote is formed. Then there is mitosis, 2-cell stage, 4-cell stage, 8-cell stage, 16-cell stage, which is the marula. Now it is 3 days now. The marula is 3 days old. On the fourth day, it enters into the uterine cavity, and within the uterine cavity, blastocyst is formed by the moving of the fluid inside, and the zona pellucida disappears. And there is an inner cell mass, and there is an outer cell mass, and a blastocyst cavity here. The this embryonic pole or embryonic end will attach to the endometrium, and this will cause proliferation of the trophoblast at this end. And the proliferation of trophoblast at this end will create a finger-like extension of cells, and these are syncytiotrophoblasts. And now the other trophoblastic cells will be named as cytotrophoblast. Likewise, embryoblast cells will show flattened inner layer of cells, which is the hypoblast. This syncytiotrophoblast is initially on one side. It will grow all